Okay. First of all, good afternoon to everyone, and thanks for the nice introduction. And I must very much thankful to the organizing committee for giving me opportunity to present our work in front of you, and especially Dr. Savary Girishan, who is we have many discussions over the phone. And thank you very much, Dr. Savary, for giving me this opportunity. And in today's presentation, I will be talking about mostly the G-scan technique because this today's seminar is mostly focused on this technique. So our lab is also working on this technique, but we are giving a new direction in the biomedical application. So I will be talking about this technique as a kind of a novel tool for various biomedical applications. So this is the outline of my presentation, where we will talk about the G-scan technique. We will start with the experimental setup and then some of the background theory. So mostly I will cover a little faster these sections because maybe from morning you are listening all these things. And next we will move to the biomedical application. In the biomedical applications, I will be talking about mostly the neurodegenerative diseases, and there are various neurodegenerative diseases. So, there are many hypotheses why these diseases are progressing. So, one of them is the imbalance of certain monoamine neurotransmitters, which is present in the neuron cells in the brain. So, we will pick one of those neurotransmitters, that is serotonin, for this talk. And there is another hypothesis that there are certain class of proteins, their aggregation actually causes the neurodegenerative diseases. So we'll pick up one of those a protein that is called alpha synuclei, which is specifically linked to one of the neurodegenerative diseases. We will talk about that. That is the Parkinson disease. So then we'll summarize, and finally I will conclude the talk with acknowledgements. So we know that I will just go very briefly in this section. So this is just what I'm showing is the experimental setup of the G-scan technique. So we know that G-scan techniques are two types. One is open aperture G-scan, other one is closed aperture G-scan. So what I'm showing here is the open aperture setup, where I will just keep the other things. The most main part I will come here. Mostly we focus the laser light, and we scan the sample on the focal region because we get the gradient of intensity here. So what we are looking for, the transmitted intensity as a function of the scanning distance. And when we are collecting all the transmitted light through the detector, we call this as a open aperture G-scan. And this open aperture geometry is very specifically looking into the nonlinear absorption component of a specific material under study. Whereas we have a closed aperture geometry that in place, like if I place an aperture before the like in the front of the detector, when I am detecting the transmitted signal as a function of the scanning distance of the sample. So there I will be discarding many signals, only focusing on the center part of the signal. So this geometry is known as the closed aperture G-scan. So in the closed aperture G-scan, mostly we focus on nonlinear refractive properties of the material. However, as we know that the materials which is having strong absorptive property with respect to the excitation wavelength, it is very difficult to remove the absorption contribution in the refractive profile. So in that case, or in even in majority cases, it has been observed that by using a single scan also, you can detect both the refractive and absorptive properties, because experimentally or practically it is very difficult to completely rule out the contribution of absorptive nonlinearity even in a closed aperture G-scan. So many people, including our group, when I was doing PhD at IIT Madras, we have shown this, that we can still go for a single CAG scan to estimate both the refractive and absorptive nonlinear parameters. So now coming back to the theory part, like the theory of G-scan is completely based on the one of the specialized subjects in physics, that is called the nonlinear optics. So when we talk about nonlinear optics, first we need to distinguish from the linear optics. So what happens in light matter interaction, when we talk about the interaction in the linear region, that means when we talk about the low electric field region. So when the light is interacting, electromagnetic light is interacting with a matter, that means the light intensity, the electric field of that light has to be very less than the interatomic field strength. That what we called as the low intensity light. So that electric field is low. In that case, what happens when there is a very low electric field light interacting with the metal, then the inside the material or any biomolecule also, you have the atoms and molecules, they will start vibrating because of the interaction. So this, when the electric field strength is very less than the interatomic field strength, so this vibration are harmonic, and whatever displacement you will have, that will be directly proportional to your the spin constant. So then we can write directly this linear expression. Polarization is directly equal to epsilon naught, then the susceptibility chi and the electric field. Now what happens when you increase the electric field strength? The moment I increase the electric field strength, that is more than the interatomic field strength, then the harmonic motion of the atomic vibrations will not be harmonic, it becomes anharmonic. And that way, the displacement will be no longer directly proportional to your force constant. That's the birth of nonlinear optics takes place. Then this equation, which was never a kind of a definition, it was just approximation, that has to be modified in case of in the presence of high electric field, where the nonlinear optics comes into the picture. So then we have this series of expression, which is the infinity series. So the nonlinear terms start appearing from the second term. This is your linear term. Apart from that, we also have now the chi2 e square, chi3 eq term. So this is related to your second order nonlinearity and third order nonlinearity. Now, if I just put it in a different way, suppose this is my linear polarization. The total polarization is the sum of linear and nonlinear polarization. And nonlinear polarization starts from this chi2, chi3 terms like that. And if I write it, the nonlinear polarization, this is my second order polarization term and third order polarization term. And if I use Maxwell's equation to derive a wave equation in term for the electromagnetic wave now propagating in a nonlinear medium. So I will have this nonlinear polarization term here, and by considering a electric field expression and using it in this equation, you can get the second order polarization term, where you can easily see a static electric field term and also a 2 omega term. So that means you are sending a omega at the input, but what you are seeing at the output is a 2 omega is a nonlinear process. That's why nonlinear optics we call as the magical branch of physics, because it creates magic. One color gives you next color in the different color at the output after the interaction with this light matter interaction. So this 2 omega term is responsible for the second harmonic generation. So that is associated with your chi2, that is second order nonlinear susceptibility. So this chi2 is basically is a tensor, it's a third order tensor. So like if you, there is very specific to structure, if you have a centrosymmetric structure, then this vanishes. Except other structures, it might survive, and even you have some surface chi2 also. But if it is a perfect centrosymmetric, then as the matrix you have the 27 elements in the chi2 matrix, all diagonally it will be 0, hence your chi2 will be 0. That means for centrosymmetric materials we will not have the second order effects. However, if I derive my third order polarization term, I will have the omega terms as well as 3 omega terms. So I will have another color light here. But as we know, to get a efficient nonlinear signal, we need to go for the phase matching techniques to get a very efficient nonlinear signal. But working with solids and having phase matching is much easier. But when you work with liquid samples, because liquids are highly dispersive, then the phase matching you cannot do it. It's next to impossible. When you are therefore when you are working with liquid samples, this 3 omega term you will never get it, but you will get this chi3 along with this omega term. That's why whenever we do G scan with liquid samples, you do not see a change of color of light in the transmitted output. And this chi3 is basically proportional to both your refractive and absorptive nonlinearity terms, because your the normal refractive index n is now changed to n0 plus eta 2i. Some people write n2 here. It depends upon what kind of unit they use, symbol they use. It varies. So you can also, this is simply as n2 only, but I just put it as eta 2. So this is eta 2i. This eta 2i, which is because of the third order nonlinearity, becomes intensity dependent. And this intensity dependent eta to I put is nothing but your change in refractive index delta n because of the high electric field. As a result of this, diameter of the beam. This is the 2D picture of a. So this picture is basically is a like a real photo of a laser light, argon laser light. So basically it is a Gaussian profile of the laser, and you have the high intensity at the center, and then intensity goes down in both sides. Now when it interacts with this, and these intensities are very high, what will happen? Now it produces a gradients of the refractive index because of this third order effect. Okay, so then what will happen? Now at the center, because the intensity is high, your refractive index will be high, and then slowly it goes down on both sides. So what will happen? We know that when the wave travels, if you have a higher refractive index, okay, then the speed will come down. Low refractive index, the speed of the wave will increase. As a result, and the waves which will distort here because of the this speed change or the gradient of the refractive index, so you will get maximum speed at the corners of the wave and slow at the center. As a result, it will converge to a very tiny spot, what we call as the self-focus. The exactly opposite happens with a negative material, negative refractive index material, and you get a divergence of the profile because of the refractive index gradient, and you see a defocusing effect. These are the real pictures of a self-focusing and defocusing from a argon laser, what I'm showing you here. Now let us move to understanding the what is saturable absorption and reverse saturable absorption. So to understand this, let us take help of the energy level diagrams from what is shown here is a ground state, first excited state, and some higher state. And we all know that with the MB statistics, the molecules always try to stay in the minimum energy configuration, and all molecules will reside at the beginning in the ground state. So just take a simple experimental concept, like you have a cell and you have an input laser light, which is now the what is interacting with the material present inside this cuvette, and you have input intensity I naught, whatever transmitted intensity I. So the moment you shine with light, some photons are absorbed, so the molecules will gain energy and go to the first excited state. And then they cannot stay longer there, and they will radiate that energy and come back to the ground state also. So this process will keep on going, and this can be very well represented by this mathematical expression, where n here is the total population, which is sum of the ground state as well as the first excited state. And if the process keep on going, what will happen? There is a possibility that we might have a 50-50 population reached. And now you see here, when you have the 50-50 population reached, that means it will look like the system attained transparency. That means as if something is nothing happening in the system, but actually the both rates are becoming equal. So that's why the populations are becoming equal. Now if you exactly whatever measuring the transmittance, if you plot it as a function of this input flux density, when the population of the upper state was less than the lower ground state, it was the transmittance will slowly increase, and when both the populations are equal, it will reach the saturable absorption state. So this is what we call as the saturable absorption in a light matter interaction, where you get a sign of beta which is negative. Now, if the process will further continue, if you keep on pumping with higher flux densities, you can just see the graph. So what will happen? Now, there is a possibility that the excited state absorption might take place. Okay. So this excited state absorption will take some of the molecules from first excited state to some higher state with further absorption of photons. In that way, the population here will start reducing. Again, you will see the coming down of the intensity, the transmitted intensity as a function of flux density, and this process we call as the reverse saturable absorption, and you get a sign beta is equal to 0. And there is also possibility that there could be a process of multi-photon absorption also resulting into the RSA effect with giving a beta 0, because simultaneously two photons can absorb and take the molecules to some higher excited state instead of this process. So RSA also involves in another two processes. One is excited state absorption, other one is two multi-photon absorption. Now coming to the theory of this kind of technique, so I will not spend much time. That's why I just summarized in one or two slides this section. So what we have here is, like, as we all know, the like you must have hard check by today. They have just given an empirical formula for analyzing G-scan profiles, where they have, you can combine up both the refractive and absorptive nonlinearity terms. As I said, CA G-scan, most of the time you have both the contributions there. If you know how to do that, you can analyze with this empirical formula, where this X is your scanning distance, and this delta psi naught are the phase shift with respect to refractive and absorptive nonlinearity. And if you are dealing with only refraction, you keep the second term, omit the third term. But if you are dealing with only nonlinear absorption, you keep the only the third term. These are just very simple, empirical formulas. And where that is useful, once you have this phase shift, you can calculate the nonlinear refractive index eta 2 and also the nonlinear absorption coefficient beta, which will also help you to find out the total chi3, because these are related to the real and imaginary part of the third order susceptibility, and you can find out the total one. And this effect basically is kind of a local effect, what we see in the optical nonlinearity. But this formula, I can also tell you this empirical equation, you can also use analyzing your profiles, even if you are encountering thermal effects, if those effects comes under the non-local effects. So I will just show you that in some of our results. So now coming to the theory of the thermal, CWG scan technique, where we mostly expect that, even I heard in the question answer, people are asking several questions regarding the thermal effects. What happens in the CW laser, when you are continuously pumping the system, the temperature and density of this medium changes in the radial direction. As a result, you see a gradient of the refractive index because of the change in temperature and density inside the medium in the radial direction. And that results in, so the contribution to the nonlinearity now, not only the local but also due to non-local contribution, which is mostly dominating because of the thermal origin. So this phenomena is known as your thermal lensing effect. So there are theories proposed based on the thermal nonlinearity. There is one model called parabolic thermal lens model by Coupeau et al. And they have proposed this, where this theta is nothing but your phase shift, what we saw delta phi naught in the Bayes expression. And later on, I also came across by Falconier's work, where they have given an aberrant thermal lensing model. So they have specifically designed this model for even people who work with femtosecond lasers, but those are high repetition lasers, which look like continuous lasers. So high pulsed lasers also accumulate many thermal effects. So to analyze that, he has designed another expression, where he has introduced a term called Q to take care of the different orders of absorption. Because you might have a linear absorption, but you might have some, multiphoton absorption. So that absorption is given by this Q. That's why I feel it is a more complete equation when you are dealing with thermal nonlinearity, because you have all the terms there, because the absorption component is also coming to the picture, and the V of Q is your thermal strength, or also it is similar to your delta phi naught, that is your phase shift. And later on, we just modified this expression during my PhD time to in the steady state condition by just removing the time part, because mostly in the CW laser, when you don't use femtosecond laser with high refractive laser, you are directly using CW laser. You can use directly this expression instead of that by omitting the time part from there. So so these are the equation, and from there this V of Q can be related to this nonlinear refractive index eta 2, or also change in refractive index with respect to temperature. Also, if you are getting the order of absorptions 2, 3, something like that, you can minimum 2, you can find that beta value also from this analysis. Okay. So now we will move to the biomedical application section. So in biomedical application, as I said, we have various diseases here. So one of the diseases what I'm focusing here is one neurodegenerative diseases. Basically, neurodegenerative diseases is a class of some diseases which has specific to happening in the neural system, like your central and both the peripheral nervous system. Because they are group of syndromes, they are basically characterized by because of the progressive degeneration of the structure and function involved in the central and the peripheral nervous system. So we have the central nervous system consists of brain and the spinal cord, and all other neurons which is running in the body comes under the peripheral nervous system. The disease list is very long. I just try to put some of them, like your Parkinson disease, Alzheimer disease, schizophrenia, obsessive compulsive disorder, irritable bowel syndrome, depression. Now it is becoming very common, even irrespective of the age, and becoming a part of our life. The way we are, progressing in the change in the society. So all these things is pose a threat to our life. And so today we will be mostly focusing on the Parkinson disease and some related project. So the Parkinson disease, basically it is a motor neuron disease, and it is mostly characterized by, because of the loss of dopaminergic neurons it progresses, and which region there is a brain we call a substantia nigra region, where this dopaminergic neurons play a very crucial role for our reward and movement. This is a 3D image of the brain, scanned image. You can see the red points are your substantia nigra, which is present in the brain. So if you just do a cross section of here, you see the substantia nigra like this, and the darker regions are full of dopamine. And in case of a Parkinson patient, to the scanning, it has been observed that you can just see the dopaminergic neurons are completely vanished. So that progresses the disease Parkinson. And because it is a motor neuron disease, so you have many motor symptoms like your shaking of hand and the body rigidity, your slow of movement, so face change and soft seas, handwriting disorder. So many symptoms are there as a motor symptoms. There are also non-motor symptoms like loss of smell, sleep disorder, constipation, mood disorder, which can individually express only then only we will come to know. But motor neurons are visible. And the current statistic says that we have almost 10 million PD patients worldwide. So out of that, 1% people normally affected after they reach the age of 60 and above. And it is more, the disease is more vulnerable in case of a male as compared to the female, because the ratio is 3:2. And life expectancy, because once diagnosed, the patient can die between 7 to 15 years. So what are the possible causes? It could be genetic and environment, and which we don't have anything in our hand. But environment, we can do a lot, but what we are not thinking right now. So, but there are other causes of the imbalance of neurotransmitter like serotonin, dopamine, norepinephrine, etc. These neurotransmitters also have, because they are mostly used for the cell signaling. So imbalance of this concentration of these neurotransmitter also pose a threat for triggering this disease. And one of the most, the protein hypothesis is the aggregation of alpha synuclein protein, which is killing the dopaminergic neurons. That is the major concern here. So we will be focusing these two, serotonin and the alpha synuclein work today. So before that, I will just tell you about the monoamine neurotransmitter, because this neurotransmitter are basically amino acid precursors, and they are involved in many, playing many significant role for us, like processing of emotion, reward, sleep X cycle, behavior, etc. How they do it? They transmit signals from one neuron to other. So they do all these activities by transferring signal from one neuron to the other. If I just show you through this cartoon, that so this is a one neuron cell, this is another neuron cell. So signal is transferring from one to another through the normal exocytosis process. So these are the basicals what you can see here. The neurotransmitters are stored here. In normal exocytosis process, they come closer to the plasma membrane and they are released, and there will be receptors in the other neuron which will catch and transfer the information. So this is the normal practice, how the neurotransmitters are involved in the chemical signaling. But in case of neurodegenerative diseases, the numbers are very low. The transfer mechanism is disrupted. So these three neurotransmitters are very popular, and they basically they control all our mood cognitive function. If you just go through each one, the serotonin, dopamine, and norepinephrine. And it is believed that imbalance of this neurotransmitter causes various neurodegenerative diseases. So as I said, for this work, we will be picking up the serotonin. So the project will be, so estimation of serotonin concentration by G-scan technique at cellular level. Now, first the question we need to ask that whether, because we will be using G-scan technique, whether serotonin possesses nonlinearity or not. Because that is the first question. If at all it is showing nonlinearity, then only we can go ahead for measuring its concentration at cellular level by bringing a model. So these are the protocols in the experiment what we have used. We used a CW DPSS laser at 532 nanometer with the maximum power output at 80 milliwatt. The G-scan setup, we have used a focal length of 15 centimeter that gives a Rayleigh range of 2.3 mm. For closed aperture, you use a 5 mm diameter aperture, so 1 mm sample cuvette, which fulfills the thin sample approximation condition. And we write our own program in Dev C++ for analysis. Also, we can use Origin and MATLAB software also you can use for data analysis. So the first one we just looked at about the nonlinear study of serotonin, where here we are going for the variation of laser power. I just wanted to see what is the effect of the laser power and the nonlinearity, if at all it shows. So the first picture what we got is, yes, the serotonin possesses nonlinearity. So it gives us a peak to valley nature, that is a negative refractive nonlinearity, negative eta 2, that is correspond to the self defocusing effect. And the theoretical models what I proposed, what I told you about, I am just showing a comparison here by analyzing the same profile in all the three models. And the comparison what you can see here, the adjacent R square values, which is showing the Sekbai model and the aberrant thermal lensing model showing the very good fit as compared to the normally the parabolic thermal lens model proposed by Kupo. Because as I said, if you have even under the non-local interactions, in many cases the Sekbai model also you can use it for your data analysis for finding out the phase and all that. So because we have shown thermal lens model, which is a slightly for basically for thermal nonlinearity or other type of nonlinearity, slightly this is a better model as compared to the Sekbai. Sekbai works very nice with respect to the pure optical nonlinearity, no doubt about it. But when you have a thermal contribution, I prefer always to use this aberrant thermal lensing model, which is proposed by Falconer and little modified by us under steady state. So these are the profiles what we got with the variation from 20 milliwatt to 80 milliwatt power. What you have seen is, as I said, if you have a contribution of absorption, sometimes what happens is like your profile is not symmetric, it will be distorted. If the contribution is from RSA, then your valley will be enhanced. If the contribution is from SA, your peak will be enhanced. So here we saw a slightly enhancement of the valley in all profiles. And so by analysis, what we saw is the N2 values, the negative of the parameters, they are decreasing as a function of the power, but the beta value are positive and increasing. So this positive value, mostly because if I have to analyze, see what is the contribution of this RSA, whether due to excited state absorption or two photon absorption. So for that, we need to look the absorption profile of serotonin. As you can see, this is the resonant absorption is falls around 275 nanometer. But whereas my excitation wavelength that is around 530. So that comes under exactly half the wavelength of the where the resonant absorption is there. So we speculate that even though we are using a CW laser, probably the what we are getting the RSA effect, because excited state absorption we cannot have it. So the probably it could be due to the multiphoton which is triggering out at even at 80 milliwatt power. So that is our hypothesis here. And then this is about the power dependent study. Then we move to the study for variation of pH and concentration. But what is the reason of going for pH and concentration study? Mostly the pH studies. The reason is like whatever I show you the cell signaling. When the neurotransmitters are stored in the vesicle, they are showing they are very acidic. But when they are released, they come to the synaptic cleft. They are in the physiological pH. So there are two pH which is actually determining their state of nature. So that's why we have gone for the pH 7.4, which is the physiological pH, and pH 5.5, which is the acidic pH. So as you can see, we have taken absorption in both the pH value to see if there any change. But we haven't observed any change, even though there is a protonation effect, which we will talk about in the result. So that is the main reason we have gone for two pH and the concentration of variation. And these are the results, as I said. I will not spend much time because we are getting the self-defocusing effect. And in case of PS 7.4, you get the RSA effect. But in case of the acidic effect, we get the negative. Yes.
Professor, sorry for disturbance. So you have 5 minutes, Professor, please.
Yeah, okay. You started 10 minutes late. You started at 3:40 actually.
Okay. So sorry.
Okay. So I prepared for 45 minutes exactly.
Okay, Professor, please.
So if you give me that time, it will be good.
Okay, Professor. Please continue. Okay.
So what we have now is actually the self-defocusing effect. And when you have the physiological PS, in the red line, what you see the data. So that we have a positive beta, but in the acidic medium we have a negative beta, except at higher concentration. And as I said, I already told you that the CAG scan is sufficient to get both the refractive and absorptive nonlinearity. But sometimes you need to cross check with the OAG scan. So we have also done that to cross check the parameters whether you are getting exactly or not, or there is some error. So here are the parameters what we observed. So always there is a, like when you increase in concentration, the nonlinearity increased, and so in different PS values the trend continued. However, as I said, the acidic medium, the strength of nonlinearity is lower. So because of the protonation effect. Now, if I look at the structure of the serotonin, the protonation cannot take place at these 2 points because oxygen is more electronegative than nitrogen. So, and also we haven't seen any structural changes in the absorption plot. That's why here or here they cannot take place because they are the ring. This is connected directly to the rings. So the protonation possibly might have taken at NS2 side, and there are several studies we talked about lowering of nonlinearity due to protonation effect, which exactly correlates to our thing. And the orders of nonlinearity what we are getting here, they are not purely thermal, because the thermal orders are around 10 to the power minus 10 meters square per watt. What we get is slightly lower than that, 10 to the minus 12, which is going towards little bit the nonlinear effect towards the saturation absorptive nonlinearity, which we will talk more in the next project. So now what is the main goal here is, now we want to slow demonstrate it to measuring the serotonin concentration because it possesses nonlinearity. So we have a platelets model, because cells are very complex. So you see a lot of scattering when you do the G scan on directly on a cell. So we thought, we observed that the platelets, which is lacking the nucleus, they are very simple geometry, and they have also lots of major content of serotonin present in platelets. So what we have done, we have just extracted platelets. Now we already have a standard curve for measuring the phase shift values from 1 millimolar to 600 millimolar, and then we taken the G scan for the in the platelets and got a value which is exactly following in the graph, which is around the concentration of 250 millimolar. So, and this is also reported that the concentration of serotonin in cellular levels, those in the vesicles, are in the range of 250 to 300 millimolar. So, though these are our preliminary results, more studies are going on to become more specific to demonstrate and measure the concentration of serotonin in a platelet model. So, we just summarize this. Like we came to know that okay, serotonin possesses nonlinearity. The magnitude of nonlinearity is higher in physiological medium as compared to acidic medium, and the concentration of serotonin is successfully estimated with the preliminary results what it shows up in platelets. And we propose that this is a straightforward strategy to probe any problem related to serotonin that is have a role in any monoenergic processes. Now we'll go to the next project, that detecting toxic species of aggregated alpha synuclei by G-scan technique. So here, first of all, because as I said, this protein is, the aggregation of this protein is involved in the one of the neurodegenerative diseases. So before going to that, first we'll understand what is protein aggregation. So normally what happens when protein is synthesized in our ribosome, so this goes for the folding process to attain a structure, because in biology structure function relationships are intact because they are very well established for a particular structure doing a particular function. So this is the usual process of the protein folding, attaining a particular structure for a function. But what happens during this translation process of the protein folding, some misfolding happens, so you get a disorder protein. That's what we call the intrinsic disorder proteins. So these intrinsic disorder proteins, they might go for different aggregation process to form a amyloid fiber. And you can ask that okay, this can be also cleared by body's own mechanism. Yes, that is true. But if when this process is not efficient, the auto degradation process, then the problem happens for the disease progression. And this method of translation going wrong to get a misfolded protein, this could be due to environmental stress, mutations, or translational errors. So here our culprit protein is the amyloid beta, which is having a chain of 140 amino acids. and it is present in our, if you see our autosomal chromosomes, that is number 4, is coded with the SNCH gene. So, and this is also present in the mostly present in the synaptic clefts, which regulates the release of the dopamine neurotransmitter, which is actually transferring to the other neuron for the signal transmission. But in case of a Parkinson disease, what happens? This transfer process is disrupted. So the main mechanism here is, suppose that protein, what as we talked about, going for the aggregation process. So this essential gene having this alpha synuclein protein, that is activated by the Parkin2 gene, which is actually naturally ubiquitinated by this alpha synuclein, goes with a proteasome pathway to get auto degraded. But if this process is not following properly natural way, then because of some defect in the Parkin2 gene, then this ubiquitination process is get disrupted, and then they form this aggregated species, and they go for killing the dopaminergic cells. That results the Parkinson. So our idea is, now we will allow the aggregation process to take place in a laboratory scale with a synthesized intrinsic digital protein of alpha synuclei, which takes place from 0 hour to 48 hours. And each stage they go for monomer to dimer, trimer, tetramer, and oligomers, late and early oligomers, and finally they form these fibrils. So there is not a single technique available now which can probe this whole aggregation kinetics in every stage. So that is the reason. So this kind of technique was proposed for the first time to monitor all the aggregation for the whole time window. And also we hypothesized that we can also identify with a particular nonlinear value for different populations. So with that concept, we have carried out the alpha synuclei project, where we have, I just directly show you all the results. So we have 0 hour, 1 hour, every hour sample were collected, and what we saw a self defocusing effect with negative nonlinear refractive index. That we got some good results the moment we come to the 24-hour, because we saw a sign change from negative eta 2 to positive eta 2. So the self defocusing effect changed to self focusing effect when the oligomers are appear, and it continued further till the 5. And in all the profiles also, we have the absorptive nonlinearity, which is dominating the refractive nonlinearity. So now to understand further what we can correlate with these results, then we looked into the literature. So we identified that actually the monomers of this aggregation species, they are not toxic as per the literature survey. Also the fibrils are also not toxic. However, this intermediate species, large oligomers, which we can compare with a gang of goons with different characters, which are very vulnerable. So they are like that. So they are already people have reported that the oligomers are very toxic. So that means our results are actually identifying the toxic species with a sign change the moment we get that 24 hours scanning of the sample. So that means the sign of eta 2 got reversed, and this can be used as a marker to identify toxic species indirectly in Parkinson patients if you have a specific parameter. So that's why this technique, this kind of technique is proposed as a tool to identify the toxic species of any aggregated proteins. And here are the results summarized. As you can see, different hour. I'm just because the table is very lengthy, so I just picked up few hours results from starting 0 to last 48 hours. We see the nonlinearity increases with aggregation time, and each population have a distinct magnitude. Okay, and we have the contribution of NLA. You can see as the Falconer proposed, the Q values are always more than 2. That is, multiphotons are involved here. And if you see the order of the nonlinearity, it is more than 10 to the power minus 10, which is specifically proposed for thermal nonlinearity. That means the saturated atomic absorptive nonlinearity is superseding the thermal nonlinearity here. And beta sheet fibrils, they are highly nonlinear, like you can see at the 48 hours. And so what will be the litmus test here? The litmus test will be if we can demonstrate this in a Parkinson patient. Now, how we can do that? Now, the idea is, like, the blood has different components. The plasma contains these proteins. This protein has alpha synuclein. So if we can specifically target the alpha synuclein and test the plasma blood, blood plasma, then if you can see the sign reversal, then that means already oligomers are present of this protein in that blood. So that person is already have the onset of the Parkinson, which is because there is no other testing mechanism to identify until unless you have some symptoms. That's why many times the disease progression happens, but people are unable to know that person is suffering from Parkinson. So the onset diagnosis, like early diagnosis is also very important, so that this disease can be controlled. And the control we will do it with a healthy people. Like you test it, you won't see the sign change anytime, because we will be having same monomers all the time in a healthy people, the alpha synuclei. So I can just summarize here. So this kind of technique is successfully monitored the aggregation kinetics of alpha synuclei for entire time window. And please remember, there are many expensive tools which is covering for studying the protein aggregation, but they are only successful in bits and pieces in different time windows of the aggregation kinetics. So the first time we have demonstrated the GSCAN technique which worked for the entire time window. And the exact protein conformation also can be identified by using as a setting parameter of a specific nonlinear magnitude for a specific population. And the sign change in the eta2 can be used as a marker to identify the toxic species, which is actually triggering the Parkinson disease. And this tool has a very useful, the technique has a very like the wide application in pharmaceutical and clinical research. And we hypothesize that this can be extended to not only alpha synuclei, all sorts of proteins if they possess nonlinearity for neurodegenerative or the other human diseases also. So, as I said, this work was is recognized by the government of India in the Press Information Bureau, and also several news media in India. And I would like to acknowledge the funding agency SCRB DST for giving me fund to carry out this work. And this work was, the serotonin work was mostly done by Sakshi and in the help of Vikas, and even the alpha synuclei was also done by her. And specifically the alpha synuclei work we have done in collaboration with the ISCB Kolkata with Professor Krishnananda and their students and his students Sumanta and Ritabrita. And because we don't have facility to run the aggregation part of the alpha synuclei or synthesize the alpha synuclei, they have synthesized it and given to us. And now I'm so thankful to other lab members. Okay.